One article that came out in the January-February 2026 issue of Coral Magazine was entitled Breeding Achievement Unlocked, Pearlscale Angelfish. That would be about the Centropyge vrolikii. Matt Peterson and I interviewed Fred Shopnitz and Dr. Cortney Ohs to discuss their accomplishment in detail. Here's how that conversation went, which helped build the story. Thanks, guys, for joining us today to have a nice conversation. We're doing some research on an upcoming story, and we have Fred Shopnitz here, Cortney Ohs. We are going to be talking about fish breeding. Matt Peterson is here, of course. He is typically our best author for the magazine. He writes some of the best verbose stories ever, where he goes into fascinating detail on the things we're doing. And your topic this time is going to be about a breeding success story about the Halfblack angelfish, or the Pearlscale.
Courtney and Fred, if you would introduce yourselves, a little bit of background and where you guys are located. I think some of our readers will know, but some of them won't.
My name's Fred Shopnitz. I work at University of Florida, the Indian River Research and Education Center in Fort Pierce, in the lab of Dr. Cortney Ohs. Oh, wait, this way. There we go. I got my bachelor's degree at Texas A&M Galveston in 2016. Then I went to work in Hawaii for the Department of Aquatic Resources, growing sea urchins for restoration outplanting with the efforts they do to restore Kaneohe Bay. Came to University of Florida to do my master's degree, which I finished in 2020. Then I worked in private industry a little bit, and then came back, and I'm now the biological scientist in this lab. Every job I've ever had has been taking care of fish. Pet store, tank maintenance, undergraduate worker taking care of stuff. So more than half of my life has been taking care of fish, or fish plus other animals.
So I was breeding fish when I was a kid growing up in the Midwest in my basement, and I had no idea what aquaculture was. And breeding freshwater fish and keeping marine ornamentals and reef tanks. And I figured out what aquaculture was through going to trout farms. And so eventually I went for a master's degree working with larval fish, and it was at Mississippi State University. And so worked on all sorts of shrimp, freshwater prawns, hybrid striped bass, food fish, catfish. I was exposed to this great diversity of aquaculture. So that's how I started my master's degree, and then just continued on working as a full-time PhD researcher, received PhD at Mississippi State, and then left and went to Southern Illinois University for 2 years, working with food fish and production on a real large scale that was occurring there. So then the opportunity at University of Florida, and I started working here as a full-time researcher, developed the aquaculture lab here at Indian River Research Education Center. There's several of them within University of Florida, and this is probably the second largest one. And the Tropical Aquaculture Lab is in the Tampa region, so we're straight on the other coast. And so I always wanted to work with marine ornamentals, and started with marine bait fish reproduction. We raised copepods, multiple different species, to help define some of the culture methods for those, how to take those up to a large scale, worked with the local producers of those, and saw it as a necessity for marine ornamentals. And so it just transitioned into that. So then I was wide open to work on whatever. So we started getting a lot of marine ornamental brood fish and trying reproduction of those. But it was a path to get here that started when I was young and breeding freshwater fish, but not knowing how to breed marine fish. So anyway, yeah. Been doing this most of my life now, I guess.
So Fred, how long have you worked with Dr. Ohs?
2 years for the master's degree, and then a little over 2 years since I came back, so 4 years total.
All right. And so during your tenure, what species have you worked on at the lab?
I did my master's degree working with the Florida hogfish, which is not a ornamental necessarily. It's more of a food fish. Right, they get huge. Yeah, right in back of Courtney. That guy.
Oh, yeah.
So,
I saw that.
So they're a commercially important fish, especially in like the Florida Keys, as a food fish. Their populations are threatened in the wild. We had the first captively raised fish here during my time. Prove that you can do it. The survival's not great, but it's totally serviceable, and there needs to be more research to improve it. Definitely could be used as a tool for like restoration efforts because, like I said, some of these populations are threatened. Um, and when I was in the lab, the Rising Tide Conservation was funding a lot of this research. So I was around other master's students, Katie McCord, Morgan Bronson, that were doing their degrees on the ornamentals. So I just kind of got to help a little bit because we're all taking care of everybody's projects. But I've worked with tangs, wrasses, Centropyge angels, anthias, other types of wrasses, a lot of stuff. The harlequin bass, that so we have more than a dozen species at any given time that is in some level of development here.
And Cortney, back to you. And I'm just trying to guide a little bit of background for people who aren't familiar.
Sure.
You've explained to me in the past that your facility and your university is a little bit unique in that you're a land grant university, so you don't have to hold things back in terms of keeping things proprietary or selling your findings to the highest commercial power, if you will. Can you elaborate on that a little bit, and then also just fill in some of the gaps of other accomplishments that have happened in your lab?
Sure. Land grant universities, there's one per state. Some states have two. And we are funded by the state of Florida, and it is our duty to do the research to benefit the state of Florida and to all of the producers that are here in Florida. So we work directly with them. We are not keeping any information. So we want to see aquaculture develop around the world, develop in Florida and all around the world. So we provide all the information to anybody who wants it. So we've worked with multiple species of copepods to start with, Parvocalinus, Apocyclops, Pseudodoptimus, and we were some of the first ones to really do some of the pivotal early work with even what salinities and temperatures they would thrive at. And so now it's exploded. At that point in time, the world, really was working with about 10 different copepod species in the literature and very little known. And now there's hundreds potentially, all within the same genus, but there's several of them around the world that are now being used for aquaculture. So that was one of the first ones. We have successfully raised Pacific blue tangs. Oh my! I have to put together a whole list.
I can rattle
Fred,
some
you
off.
want to jump in with
Cuban
me?
hogfish, yeah, Cuban hogfish, Florida hogfish, golden trevally, harlequin bass, half-black angels, hutchii, anthias, copper band butterflies, candy hogfish. It's a two-spot hogfish, little guys, and that's just in the last 5 to 7 years.
Banded butterflies, spot fin butterflies.
Banded butterflies.
I think reef butterflies
Yeah.
too, if I remember correctly. It's like the only one you
Yeah,
haven't
reef
done.
butterflies. Yes.
Yeah, four eyes, which I keep telling you, stay away. I want that one. I want to do four eyes. All right, so I mean I think that's a pretty good overview. But I want to one more thing, because I think right before we started the recording, I think one of the goals that I laid out for the article that we're putting together, based off of all your research for the Halfblack, is we want to kind of reinvigorate home breeding and at least show people that it can be done at home. And you had kind of presented this to me, the Halfblack, is this is something that at this point in the world, a home breeder could tackle. It won't be easy, right? Fred, would you say that this is easy or a challenging project?
Yeah, I mean it's a challenging product or a challenging project, but if you have your home aquarium set up to do breeding activities, it's not like a display tank with a hang on the back filter and whatever. And if you don't have a closet full of algae and copepods, which a lot of hobbyists that read this magazine actually do have a setup like that.
It's going to be more difficult than breeding clownfish, of course, but I think um, it's within the reach if you are.
Definitely.
Yeah, and
Yes.
so because I come from the world of marine breeding in like the late 2000s and the 2010s, where everything was just kind of exploding, and the world of marine breeding was, any home hobbyist had a fair game at breeding, like a new dotty back, or like I was the first to do the harlequin filefish, and I'm still frustrated that no one else has replicated that work. Um, but then I feel like in the mid 2010s, it really kind of shifted where a lot of the breakthroughs started happening with pelagic spawning fish, and required almost more commercial or academic scale resources. And Centropyge angels were kind of the exception a little bit, that you could still maybe accomplish it at home, like Kathy Leahy with the Coral Beauties, where she did it in her basement in St. Louis. Um, but since then, we've refined a whole bunch of that, and so I think today is a great opportunity to use the Halfblack as an example of saying this is still something now, with another 10 years under our belt, that Fred and Cortney, you can take a modern approach and lay out a protocol and say, here's how you would approach it now. And then before we dive into that, and some of the questions I have, because we're not going to lay it all out here today, that's not our goal. I'm going to ask some questions that weren't covered in the initial notes, and so we'll have some teasers about what's going to be in the ultimate article. But I want both of you to speak about, the person who's maybe 15 or 16 right now. How would they go about coming to the university and maybe one day winding up in your lab? I just want to, like, if they get inspired by this, how do they wind up being in your lab, or one day being one of you guys? How does that look?
So locally, there's volunteer opportunities. Um, Typically, you need to be 18 years old, and those opportunities are available. We bring in interns all the time, so there's different agencies that help recruit them, and that just come to different labs. So that's always a possibility. University of Florida doesn't have an undergrad program in aquaculture directly, but there's a lot of the courses are offered for it, and it's part of the fisheries and aquatic sciences. So there are possible options there as an undergrad. You still take some of the same coursework and stuff, but it's looking at more diverse than just aquaculture itself. And then within graduate studies, then you're doing research projects in one of the labs and taking all the courses. Most of the courses are now offered online as well. So we have a wide-reaching online presence in teaching our coursework. I have 68 students in Introduction to Aquaculture right now, and they're scattered all over the whole world, and they're half undergrad and half grad students.
So wait, are you
Um,
are you telling me that I could come back to school right now and be one of your students and and get my master's degree from you and set up my lab in the basement and do research under you?
well, I don't know about all of that. I mean, but it's not impossible. And so a lot of people with the skill sets that they're developing doing this at home are just a easy transition to a research scale as well. So they need to, learn the mathematics, the statistics, and the things like that. So yeah, it's not impossible at all. Going from the demersals, the clownfish and the dottybacks, stuff like that, then going, one thing that we do is we collect eggs from every single tank, and it's the first thing as it exits the tank. And so as they're pelagically spawned, then everything goes into a collector, and we have that all defined, the screen sizes and everything that work perfectly. It's through the University of Florida EDIS documents, and you could do some sort of a modification on those for a home aquarium as well. And so lots of people that have an aquarium don't know that spawning behavior is occurring because the filter basically sucks them up. And so that's the first step. That one thing we check every single tank in the whole facility um, every single morning and first thing to see what has spawned. And so that is the first thing that you could do, because I believe a lot of fish are breeding and spawning within a tank and potentially fertilizing, and you have no idea because they're sucked up by the filtration system.
That part's true. I know it was Matt and I have had conversations about why I've never tried to raise fish, and he figured it out really quickly as we talked, because I never have pairs of anything. You gotta have two to make more, and I always get one of this Nassau tang, one of a, I mean, everything's just one, and the cross pollination just doesn't really happen in my reef tank too often. But I'll tell you, there are people out there that are more aware of what's happening in their tank after lights out or during the final moments of lighting. Like, for example, a lot of people love the mandarin fish dance, that where they two come together. But what was really interesting, caught my eye recently, because you never see this. One photographer finally took a photograph of the eggs as they were released, because they always film the fish and then they follow the fish away and they never show the spawn. And this was the one time you had like a perfect macro shot of what was just released before the flow dispersed it. That was a really neat thing to see. What made you pick this fish?
When I was at Seagrass and we were looking at different fish that they had imported and looking for quality broodstock, they had several of them that fit the bill perfect. They were large individuals, and we were looking for ones that would be broodstock in a shorter period of time than others. So they were not on our list of what we were collecting, but when I saw them, I'm like, those look really mature, larger individuals, and they're strong. And so we just picked those out of the shipment directly then.
So Fred, why don't you walk us just a very high level kind of tour, just a summary of what you did, because the Halfblack project was your project, correct?
I think it's like shared between kind of everybody here. We have so many different species of fish that very rarely is somebody assigned to one species. I would say it was a collaborative effort between myself and Jose Ordonez Montoya, who was a student here and then came on as an OPS worker. Andrew Palau is a technician that we have, and we've all sort of collaborated with Dr. Ohs and some of the other workers here to figure out, this isn't working. Oh, it worked with this thing, and like little minute changes that we can make. So the Halfblacks that produced the very first spawns, I think they were in the facility in like 2020, right, Cortney? Is that when you got them from Seagrass? I mean, they've been here a while, and we've had many other Halfblacks that have not paired off nicely and have not made it. So going back to what Dr. Ohs said, like, broodstock selection is incredibly important. If there's ever fish that a diver is seeing that's like a pair of butterflies and they're going around in the wild together, getting those two exact fish is worth so much more to the type of research that we do than getting 10 random ones and trying to figure it out and watching these beautiful, expensive fish beat the heck out of each other because they don't like each other. So with pairing up Centropyge angels, that was a bit of a challenge, but these two were a pair for a very long time. They spent some time in 30-gallon aquariums. They spent some time in 75-gallon aquariums. As we were seeing increases in spawning success with other ornamental species that we have, like the harlequin bass, long-nosed hawkfish, we sort of followed some of those changes. So when they were living in 75-gallon tanks, and I think this is important, they kind of tried to spawn a little bit. Because we have these egg collectors, we were able to notice like 10 eggs, 2 eggs, an egg that gets caught in this collector, which means that maybe they were trying to do a spawn rush and then they aborted it because they couldn't quite make it. And that's something that we've noticed with other species where you change the depth of the environment that they're in, because they're not living in 24 inches of water. They're living 30 feet down. And I found plenty of my fish, on the ground because they didn't know that the air's right there. and they they jump right out. Everybody's had gobies or dotty backs or whatever that jump out. So we moved our fish to a much deeper, much larger tank, a huge amount of space investment for two little fish that are this big, but only within, I got it pulled up right here. Let's see. They move in May of last year, and they begin spawning literally the next day, and then we get our first fertile spawns about 6 months later. But it was after 4 years in other sorts of conditions where we weren't really seeing any sort of spawning activity, and we were also changing their diets. Certain fish, you're okay to just feed them a very basic diet, even like a standard pellet diet is fine to keep fish alive if they're just in your display aquarium. But if you want these fish to be producing good quality eggs all the time, they need to be taking in high quality fatty acids, antioxidants, vitamins, because on the reef, they want to be spawning, potentially as many times as they can. And you need to constantly be restoring that good energy. So there were some diet manipulations, as well as just the physical space that they were located in that I think really, really aided in the ability for these things to actually produce after years of being here. Because we found out, now that we have F1s, just one quick thing, they don't need to be 5 years old. I have fish that are 9 months old that are spawning right now.
Awesome.
So those fish were capable of reproducing. They were capable of reproducing as soon as we got them from seagrass. It was about figuring out the conditions and the diet that they needed to actually get going. Because that's always a question. You take these things in. I don't know how old a copper band butterfly needs to be before it breeds. Maybe it needs to be 5 years old. You don't know because nobody's done it.
Yeah.
So I wanted, that was one of my end follow-up questions, but I'm going to jump back here because in your initial pair, your notes say there was no dimorphism at all. So one of my follow-ups was, so they were the same size, everything exactly the same?
I would say so to the naked eye very quickly from a distance, because when you have your little 50-gram fish, we're not taking them out and cannulating them or doing any sort of really in-depth analysis. Now, I will say, if you look at them now, it does seem like one's a little bit bigger and a little bit darker, but when they were agreeing with each other but not in spawning form, when they were just sort of living as a pair but not doing anything, you really couldn't tell the two of them apart.
Okay. And then overall, just how large was that initial pair? Like, just rough ballpark in terms of size. Were we talking 3 inches, 5 inches?
Yeah, like that, like the standard one that you would get at the your local aquarium store. 3-4 inches or so.
All right, but now contrast that to your 9-month-old.
I think if you measure the tail, like that.
I don't know. I'm saying more 2 inches.
Oh, okay. But then what about your little precocious 9-month-olds? I mean, how large are those spawning at?
The biggest fish in there when spawning started was maybe 2 inches.
Yeah. All right. So smaller. All right.
I have a question for you about the eggs, only because I've been indoctrinated recently by Matt to raise some fish, and I did not know that the eggs are hard. I thought they're gelatinous, so the fish can swim out. At least that's what mine is. Are the eggs you have hard or gelatinous?
Well, describe them. Describe the eggs of these fish.
Yeah, so the eggs are firm enough that you can pick them up and not harm them. When we use the egg collection device that the facility has on every single tank, it's just a bucket with some mesh in it that has water routed into it a certain way, and you just tip that thing. In fact, I got one right over here. You just tip everything into this, and then it all collects on the screen here, and it's hard enough that you can just take a thing of water and wash it down and then collect it in a cup, and then you can see, all right, what did I get today? So
you're not having to handle them. That makes sense. I was just curious if you touched any. I just wanted to know what they were like to the fingertip. I was curious.
Kind of like Orbeez, like really, really, really small Orbeez.
Okay.
And how large are they?
A little under a millimeter for that species, but I mean, some of the eggs, Courtney, you can speak to this. It's totally species dependent. Uh, and it doesn't
Each
necessarily
one.
mean that a small fish has small eggs. Some of these big fish have very small eggs, and some of these small fish have quite large eggs.
Okay.
Well, Mark doesn't know the fish that he's rearing still. He hasn't quite figured it out. That was a whole side project that had someone send him some eggs, and eventually he'll figure out what he's got. And they are very small fish, but they lay very large eggs, so it's kind of
Yeah,
funny.
and so I caught them. I was doing a water change, and I was siphoning the bottom of the tank. All I want to do is raise the fish. I did not plan to raise babies. But these fish are making more fish. And so then I felt guilty. I got this bucket full of salt water and cyanobacteria and all this crap in the bottom, and I just know the eggs are at the bottom. And so I said to myself, well, I'm just going to go ahead and I'm going to pour them through a breeder basket, which is sort of like your strainer thing there, and hopefully I can catch these eggs. And that was my goal, was to maybe save some lives and not be a jerk. And I saw like one on the bucket, and so I thought, oh, let me grab it with my fingertip, and it was really hard, like a piece of glass. And I was just like, whoa, I really didn't expect this. And I was kind of like, I mean, obviously I didn't mush it, but I was just like, it has serious tension to it. And I thought, how does a fish even get out of it? Because it's so hard, and because I've only seen clownfish eggs. Well, I've seen different eggs over the years in my tanks, but clownfish, they just looked soft, wiggly. I don't know. I just assumed it was gelatinous. I've never raised them, so I don't know.
They're rigid.
They are. Wow. Okay.
So,
Sorry to take you off track.
No. We're having a conversation here. Fred, once you collect these eggs, you're stocking them into rearing vessels. What kind of vessel are you using there?
In our larval room, everything is fiberglass tanks. They're cylindrical. They're usually painted black or maybe black with a white racing stripe. They're as small as 3-4 gallons all the way up to ones that are about 100 gallons. So that has to do with how many eggs and what your survival anticipation is going to be, like what you think is actually going to live in that vessel, because they're going to spend the next 30 to 90 or 100 days in that thing. So you have to plan around not only just the initial hatching of these things, they're very fragile little creatures. So that's their new home for a long time.
Are you recirculating the water through those tanks at all, or initially not? Has there been some protocol that's become standardized? I know early on, I was trying to come up with a black round tub system, BRT. It was the common acronym that we would use, and I was using like plastic muck buckets, and I think it was Dr. Andy Ryan up at Roger Williams who really pointed out that it was much easier to sanitize the fiberglass than these black high density polyethylene tubs that I was able to get. I think it's polyethylene, but can you talk us a little more about like what the preferential size would be, or if someone was trying to do this at home, what they would use, or where you would lean them towards, if you have any ideas?
I would say a couple non-negotiables are very intense biosecurity. If you're trying to just raise larvae and the types of food that you're going to feed to the larvae, that's all you want being introduced into that system. You do not want your little starfish getting in there, or sponges or anything else, because you're about to create the perfect soup environment for every larvae and every small little critter. So when, something like a sponge or something like that gets in, it's going to be competing directly with your little baby fish. So it's helpful that these eggs are larger. They are firm and rigid enough that you can put them onto a strainer and separate them. So in terms of like a vessel size, something that is cylindrical, I would say, is best, because the way that you want to introduce flow into the tank, you do not want dead spots. You don't want corners. And there are plenty of places that can raise things in 10-gallon tanks, 75-gallon tanks. But if you want to make it easier on yourself, you want to be able to have, a circular tank and take advantage of just the hydrodynamics of that. And Mark, you were asking, like, Is there sort of like a standard, like a protocol? I would say a good start is every day having some amount of an exchange of clean water, whether that is a batch of water that you have made up if it's a very small scale, or having some sort of clean recirculating system. Everything that leaves our larval tanks gets screened down through a filter to either 25 microns or 1 micron through a bag filter. There's a protein skimmer on there. There's UV sterilization on there. Nothing should be coming back through at all in a perfect situation. And every day they're getting new clean salt water, and they're getting an addition of new clean nutritious feeds. Because if you're feeding something like a rotifer or a copepod that hasn't eaten for a while, it's just losing nutrition over time. Your 3-day-old rotifer that's floating around in the tank, even if the larvae strikes it and eats it successfully, it hasn't. That rotifer is 3 days less nutritious than when you put it in there.
How do you water change out of the system without losing rotifers and baby fish? How does that work? How do you screen, or do you water change into a screen and then dump the screen back into the tank?
All of our tanks are plumbed with a hole in the center, and that has a mesh standpipe of a specific size based on the type of fish that you are having in there and the types of feeds that you're trying to retain or get rid of. All of that routes to an external standpipe, so that determines the level of the tank. So that's why you can have a mesh standpipe in there and not have all the water go down the drain. And depending on how complex you want to make your plumbing system, you could take a very large vessel tank, like a 100-gallon cylindrical tank, and you could externally plumb it to whatever height you need it to be. So
you could just add more water to the system and then remove some excess from the sump, I guess. I'm just trying to think, how do you not hurt the livestock in the breeding tank or in the raising tank?
So the way we do it, the way a lot of facilities do it, just based on feed availability, is the larval tanks are static during the time of day when we're feeding them, so there's no water exchange going on, and then your feed concentration is as high as possible. And towards the end of the day, you would turn on a light flow, which is enough to not push the fish down the drain, that have a little bit of agency and can determine whether they want to swim, but it is high enough to push something like, a 200 micron sized rotifer down the drain. And balancing that flow rate to that screen size is a very important, and just minute little changes in that or in the rate of aeration can wipe out a larval tank if you mess those things up.
Thanks.
Screens will allow the live feeds to exit and retain the larvae. So having the right mesh size throughout the day is important. And we've done very little to no exchange during most of the light time hours with live algae present and the live food organisms, and just give them a perfect feeding environment, and then flush the algae out and the uneaten rotifers and copepods at night by switching a screen size and turning on the flush. So
that raises a great question. So you guys are using a photo period that's not 24/7 illumination and feeding?
Correct.
And these tanks
we
have
do
no
not
windows.
go 100% dark.
You only have the view from above, right?
We have lighting above each tank that's controllable, and we have used a small light in the corner as kind of a moonlight for it to never be completely dark all night long.
And what is the typical photo period that's worked out for these Halfblacks?
It's around like a 12-12 photo period.
12.
Yeah. A lot of the species that are in the marine ornamental trade are equatorial, and we try to just sort of stay within that range. If you were raising something like an Atlantic cod or some other, like, far north or southern hemisphere thing, they might do better with a much different photo period.
Okay, so one of the questions that came up as I was going through the notes, you had mentioned that you start off with T. isochrysis as your your green water that you're stocking in, even though it's brown water, I guess. And then you add in rotifers. You're using rotundiformis. At what point in the stocking, when you go, you're starting with Parvocalanus crasorostris, I'm assuming nauplii, and then you're adding in the rotundiformis. At what point in your feeding cycle are you starting with the rotifers?
With your small larvae of whatever species, certain larvae seem to have more agency about what they can and can't do, what they can and can't strike.
Mm-hmm.
Something like the half-black angels, because all your pelagic larvae are starting out without eyes or a digestive tract. They're just a spine that floats around for like 2 or 3 days, and it needs to have enough nutrition in its yolk to even grow their mouth and their eyes and everything. So species that we've successfully cultured in the past before I was here, like the Hepatostang, and they don't have a lot of feeding success on faster prey items, but they need them. The Centropyge angel, they seem to actually be able to target and almost do a little like strike motion very, very early into the life cycle. So that's a long way of saying we do rotifers and copepods at the same time, because I'm not too worried about them overexpending energy trying to catch copepods, because they seem to be able to catch them pretty well. And then the rotifers are sort of like just easy prey. So why not have them there as well?
Yeah.
Yeah.
Are you enriching the rotifers with anything specific?
We've used different enrichment products. Products from Reed Mariculture and Inve have all worked very well. We've not done any sort of side-by-side contrast. That's can be incredibly important, because all of these products are different in the amount of highly unsaturated fatty acids and vitamins that they have in them. But we've just sort of used what we had on hand. But yes, they are getting enriched.
And so pretty much from that standpoint, your notes are very, very detailed about the different runs, the number of juveniles you settled out, the timing of everything. But one of the things I noticed, especially looking at the first 3 successes, was you went from an initial survival rate on the first run of success. It was 0.6%. Then your second one jumps to 1.89%. And the third one jumps to 5.2%. And as I read through it, there's really nothing different. In fact, I think it basically says you did the same thing every time. And so, of course, the scientist in me goes, well, what? No, something had to be different. What changed between runs 1, 2, and 3 to have basically an order of magnitude difference in success? Something was not the same. What happened?
I would say it's the little minutia of figuring out flow tank dynamics and cleaning measures and stuff like that. When you get a new species for the first time, you are babying that, like, no pun intended, they're little babies, but like you're observing every little thing that you can about them. So yes, all of our runs were pretty much the same of addition of live algae for the first like 30 days or so. Yes, they got rotifers. Yes, they got Parvocalanus nops. But just being able to notice to what degree they can tolerate the flow rate of the flush. Maybe it was on for a slightly different amount of time, or for the initial hatching stages, we may have tweaked a little bit of 48 hours of consecutive flow before they get actually fully formed and able to feed, versus 12 hours or versus static. So those parts weren't in the real notes. They're not as measurable. And then also, I think it's important to point out, we didn't necessarily need to change anything else. Once good quality eggs start coming out of the fish, their first couple spawns might be fertile and in large numbers, but there's just some little bit of nutrition that's missing in there. And once they push past those, then they've got really high quality eggs after that point. This is not an uncommon trend that we've seen where we can have a fish even spawning fertile for 6 months, and then something changes about the diet, and now this fertile egg is point something 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 microns bigger, and the larvae is, a tiny bit bigger, or gets to a developmental stage in 9 hours instead of in 11 hours, and that's just a huge uh, what's the word I'm looking for? It's very impactful to their survival, because these things are starving to death in a literal matter of hours, like less than 24 hours potentially. So if they can reach a developmental milestone an hour earlier, then that's a full hour of feeding that they're able to accomplish and store into the next day of growth.
So I'm hearing two really interesting points in that response. And one is like almost the intangibles of the quality of your eggs and the brood stock, and things that are really hard to pin down. And the other is something that I don't want to make this about myself, but it reiterates something that I say a lot, which is the intuition that aquarists get from experience. Those intangibles that you only notice through the repeated act of doing and observing, that there's no teaching of it. You have to do it. You have to be there watching, making those subtle observations that you almost intuit, that wisdom you get just through doing something again and again and again. That I can tell you what a stressed out fish looks like, but until you see it and start to recognize it, where 10 years in across the room, you notice there's something wrong, where your first year as an aquarist, you don't pick up on that behavior. So it's just interesting to hear you relate it back that, hey, something about those flow rates, or the way our husbandry, we refined our husbandry just by observations and going through it. And then it also, something you said earlier leads into a question I had, was I noticed that in the end, your notes, you ended your notes to us with 7 successful runs at the time. And I think there had been more since that, so we might need to swing back at the end here after we do our interview, just to see if we need to update this at all. But you had said there were 31 larval culture trials, and you had 7 successes. So 7 out of 31. I want to ask, what about the other? What about all the other ones? What about the ones that didn't make it? What would you say about those? All the failed runs.
One thing that might be a bit of a damper on home aquarists that want to attempt this, but it's something that they need to know. Every successful run had a steady supply of copepod nauplii for a very extended period of time, to the point where we basically never on purpose tried to raise them just on rotifers, because we knew they would die. But there were times where we didn't have enough to go around, because when you have 15 active larval tanks of various sizes with various species of fish, you start to ration out and say, hey, this tank doesn't look so hot, and that they went down since yesterday. I could tell the population's decreasing. We're just going to call it, because, if we go and baby this tank, maybe we'll get a few fish out. But these young guys over here, they're ripping. They're great. They're going to get the ration of copepods today. So definitely that's been a huge factor. Some of the just immeasurables of just egg quality, like I said, just because they're getting really good quality food, every spawn is not created equal. You can go based off of overall fertility percentage, and I think that's something that over 15 years of spawning here, even not just marine ornamentals, but with the marine bait fish that this lab has worked with, and other things like that, that's a good metric to go off of. So if you only have 25% or less of your eggs were fertile, it probably is not because the fish messed up when they were doing this and coming together and spawn. Sometimes it is, but it could be that there's something wrong with the gametes of those things at that exact spawn. So, I could go back and give you a more detailed look at some of those successful runs, but I mean, we're talking about we pull out our eggs, and it's hard to find infertile eggs. Where when you've got 95% overall fertilization, you can be pretty sure that run is going to do pretty well.
Nice.
So it's kind of like, if you're a home hobbyist, you want two things to come together. Maybe you have to pick and choose your battles, pick and choose which runs when you're going to do a run.
Yes.
You want to make sure
Yes.
you have a good fertile, high fertility spawn, right? Is there a magic number that you would just say based on either of your experiences, like, hey, if it's below this percentage, don't waste your time?
So, I mean, we're conducting research, so we continue to carry out fish that didn't have as high a fertilization as others. But I think I'm guessing 25% would be a bust number. But any fertilization for some of these species is good, and so we've ended up raising like one of an angelfish that we had very low fertilization with.
Yeah,
So
but if you're gonna,
research
but if,
purposes is a little bit different than doing it for a larger scale kind of thing.
but if you want to have a reasonable chance at rearing, if you're gonna invest all the effort, getting just one fish out of it is always kind of a, I don't know. It hurts. It hurts if you're
Yeah.
gonna like kill
Yeah.
yourself for 2 months to have
Yeah.
one fish at the end. That hurts. I mean, I know I did it with all the Harlequin files. Like every good successful run was one fish, and then having 50 at 35 days and leaving to go talk, and I come back and they're all done. It's like, oh, there goes my best run ever.
Yeah. Yeah.
At that point they all should have made it. And then to move and lose the brood stock kind of killed me. But so maybe there's not a magic number, but the higher you'll. Well, okay. Let me before I go to the second part. How do you test fertility? A real quick, just sidebar on that. How do you check fertility? And a pelagic spawner.
We collect the
Fertility of
eggs.
spawn. Yeah.
Now, how do
We
you know
collect
that they're 95% fertile versus 5% not? How do you know?
So in general, fertile eggs will float, but that's not always true. So you can do that on a large spawns. But we take samples of it, a homogenized sample, put them on a microscope slide, look at them, and count percentages of the fertilized eggs.
Okay.
You can tell
Okay.
the difference if you're looking at them under a dissecting scope.
All right. And then
It's
so
real clear. You can sometimes see like embryo misdevelopment and some of them, that kind of thing as well.
Wow. Yeah, I mean, just to that level, you can just look at it and say that's not going to be a viable fish.
well, even
Yeah,
like
under
your
a
eggs, Mark,
microscope, yes.
Wow.
like your eggs and your fish, we were looking at, and we're like, all those white ones are, yep, they're no good.
Yeah, you told me
Yeah.
the white eggs. That's why I was wondering if you had something
Yeah.
similar, like an egg is
Okay.
white versus an egg that has some little worm moving around inside it.
And then the other thing is really, and we're not gonna discuss this in the article, and I don't want to get into a lot of it here, but it really is, it really seems like Parvocalanus crasirostris is the magic bullet for so much of this, and it's not an easy copepod to culture. But has there been any breakthrough as of late in terms of making it any easier to culture that copepod in general?
We have pretty well-defined methods that work real well. We use stainless steel screens to separate them and have automatic collectors that will collect only the smallest nopuli and go into screen collectors. So we come in the morning and we have them all ready. So we've engineered our systems for those.
Is there information available to the public that can replicate this methodology?
Yes, there is. University of Florida EDIS documents have several of them in different screens, and we're happy to share the different screens. We found that stainless steel screens have a perfect mesh, and they don't really attach so much flock to them, and so they stay cleaner and they won't clog as much as like a Nitex screen will. So they work really well with the copepods, and they're basically just pushed through. The nopuli is pushed through a screen with flowing through out into a collector, so only the nopuli are going through.
So, and I know this kind of came up in another conversation. I think it was talking with Till Theus and Adam Heinrich
Sure.
when we were talking about Serranus tortigarum, where I think they came up with some number. I think they said 900 gallons of Parvocalanus copepod to culture like 30 chalk bass was the kind of their math. which seemed insane to me. It was like, well, there goes half my fish room just to copepod culture.
Yes.
This is like, I don't
It's
know if that's
crazy.
realistic. But what would you put it at? Like realistically, I mean, what kind of scale of copepod culture am I going to need to support just one larval run of a Centropyge like the Pearlscale? What am I going to realistically need, food culture wise?
You gonna jump on that one,
I,
Fred? That's hard.
I
A
can, I can, I
barrel?
can break, I can break this.
I mean,
Yeah,
ballpark it. But I mean, is it really that bad? Is that really what I'm looking at?
I think a barrel or like a large trash can, like a 32-gallon brute trash can with an airlifting collection device, which is described in one of the publications that Dr. Ohs was talking about, um, and then with all of these things that you're trying to do, you're compounding work by having to maintain other organisms now to
Yeah.
sustain your little baby fish. So you're going to need to set aside some of your very precious copepod nauplii and grow them up into your next generation of adults, because at a week, 2 weeks, 4 weeks go by and they're not going to be producing very well anymore. So you're now taking care of that, and then you're growing your live algae to give your copepods the best nutrition
Yep.
too. So yeah, it's a whole walk-in closet dedicated to something. You're going to have more here, let me put it this way, you're going to have more live feed space probably than you're going to have larval and brood stock space, unless you have a really, really, really big display aquarium.
Yeah. Oh, go ahead, Matt.
I
Sorry.
well, look, I mean, I think I realized very early on that like you do this, and then also realistically, you need a lot more grow out space too. If you are successful at rearing
Yeah,
larvae,
once
to
you
hold
actually
on to
get
these
them, yeah.
fish, once you have larvae, there you got to hold on to them for a while to grow them up before they can go anywhere. So, the amount of space for broodstock and larvae is very small compared to just holding on to grow out until you can move them out into the world. So, I feel like it was like 10% of my space was broodstock, 10% was larvae, and then 90% was grow out. And then when it came to food culture, that was a whole nother dedication, much larger than I needed for my broodstock and my larval systems, just to have enough for rotifers and algae. And I remember going, man, I hated growing phytoplankton, and I didn't need it for a lot of what I did. So until I was going to pursue parvicolanus, I was like, I could do all this stuff with pastes and frozen algae. Until I need parvicolanus, I'm no longer growing algae. It was just too much work. It was 8 hours a month was my live algae culture time budget just to grow algae. That's a full day's work
Yeah.
every month as a hobbyist just to grow algae. It was like, that's a lot of my time. So I'm not gonna hide it from anyone. Just me self-assessing my time. It is a project. It's a lot of dedication, and I think you have a team and you share the workload. So it is quite an accomplishment for someone to do this as a home hobbyist. I'm not going to sugarcoat that for anyone who's watching this, that this is going to be a project and a work, but also I think a real sense of accomplishment if someone does this in their basement. It is a challenging project, but very rewarding if you do it. And whether you do it with a Halfblack or any angelfish at home. Let me ask you this, Cortney, because I think you guys have done pomacanthids in your lab, or no? Or pomacanthus, or some of the larger angels, are they any easier, or
No.
no?
They're
Okay.
not any easier, that's for sure.
Are there any pelagic spawners that you would put as easier than any of the angelfish?
For sure.
Oh, okay. Fred,
Sure.
you want to jump on that?
Jump on.
That
Well,
you
I
could
mean,
do with the same tools that you would apply to the Centropyge, that might be a step in. Like if this is maybe just a little bit beyond, but you could learn the techniques of the algae, the copepods, apply it to a different species prior to tackling a Centropyge. What would you say you would start on?
um, well, there are certain like demersal or like they're fish like dottybacks that will lay eggs in like a big like jelly sack. So they're not pelagic spawning fish necessarily, but that's you can apply all of these same techniques to fish that are actually going to survive well, and that can allow you to sort of hone those techniques. Yes, you don't need those things for a clownfish or for a dottyback, but you will find that you might get 10 times better survival if you're actually able to provide them with that sort of an environment. And then in terms of readily available pelagic species that you could pick up at the aquarium store, Centropyge are a pretty good start. There are some ornamental species that are just starting to come into the trade. Australian stripies are one in particular that I know Jonathan Foster at Fisheye has marketed to public aquarium, where the survival is like 10 times better than Centropyge angels. And those were marketed through Jonathan Foster, but I mean, I was the one growing them, but whatever. Yeah. That's a side story.
Oh, okay. So okay, so like, you know, stripies. What about like I don't know, any of the Anthias, like or any of the. I was thinking if there's any other, like mandarins, obviously would probably fall into the easier camp because they have a shorter pelagic life duration. Is there anything between a mandarin and a Centropyge that might have a shorter pelagic phase that might, as a pelagic spawner, might be a bridge to get to the Centropyge or the pygmy angels? I'm just brainstorming here.
I'm trying to. There are ones that the larvae themselves might be easier, but the home setup is not conducive for it. You mentioned an Anthias. If you had 25 Anthias in your home aquarium and they were all spawning. I remember when I worked at a pet store in Chicago, a guy bought 12 Bartlett's Anthias and he's like, what the heck is this? And he was getting spawned. They just started spawning in his tank because there's 12 fish all together and they're all happy. But that's not most people's aquariums. But Anthias and grouper species, Serranidae family, they can have a higher survival rate per 1,000 eggs than something like a Centropyge, but you're not going to have those generally in your home aquarium setup unless that's your big project. If you want to get one male square Anthias and 5 female square Anthias and have that be your whole tank, then those would probably be easier to grow if you had a sustained supply of fertilized eggs. But if you just got a pair of sunburst Anthias and they're this big, then you're only getting 300, 500, 1,000 eggs at a time, and just your total number of eggs is not going to be conducive for a home grower.
So someone like Mark, who has a 400-gallon reef tank, if he modified his tank to put a shoal of Anthias in, it would work. But that also, I noticed you had long-nosed hawkfish in one of these broodstock tanks for the notes. I'm just brainstorming here, but you mentioned the groupers, and then you guys had done the harlequin bass.
Harlequin bass, tigrinus,
Would that be a species, or any of those Serranus species might be a good target? I tried to do the annularis, the orange backs. I had those spawning in a very small tank for a little while. Mini groupers. I mean, any of those little mini groupers might.
Chalk bass, species like that.
Yeah.
I think those would be as easy, given the right setup. Just a little throwback to the harlequin bass. We had multiple populations of those fish, and it was a lot of infertile eggs over and over and over and over. 10,000 fertile eggs, infertile eggs a day, and it was a dietary change that made all the difference. And you could see literally the larvae when they hatched out were larger and more robust, to the point where I almost thought we mixed in a different species of eggs. I'm like, these guys are huge. This is the wrong thing in here.
What was the dietary change, if you don't mind me asking?
Just higher quality fish meal sources. Let's say it like that. So, salmon, high quality, like tuna, mackerel kind of species, and understanding that more fat is not necessarily better. It's your source of your fat, your freshness of what you're using. I don't know any off the top of my head, but I'm sure there's plenty of like prepared diets that would fit a lot of this stuff. I know we use a lot of Larry's Reef Service, or the Fertility Frenzy stuff, or just products by Larry's Reef Service. Those have been very, we feel, crucial in getting multiple species of ours to spawn. But, you might think you could just throw fatty krill at these things all day alone, and you think, more fat, more, egg production, but you need to have, your DHA, EPA, ARA covered and then antioxidants. You don't want to forget about beta carotene, carotenoid, different types of carotenoids, astaxanthin. So a lot of the stuff that we feed our broodstock is like a custom blend, and lots of facilities have their own little tweaked versions of them, and they change as people learn different things. And I'm sure there are some posted recipes. I know. Here's a good place to start. There's a paper by it's out of Hawaii Pacific University. It's out of Oceanic Institute. I don't think Dr. Callan's on the paper, but it's a paper about growing Centropyge angels. It's a paper about flame angels, I think, and they outline the like omnivore diet that they were making for them. And it was a gelatinized diet that you needed to make with like 7, 8, 9 different ingredients, and then you blend that up into like a jello almost, and then that was what they fed out. And that was like the basis of some of the diets we use here, some of the diets that they still use at Oceanic Institute and other research institutes that are working with marine ornamental fish.
I think broodstock nutrition is critical, and there's like Fred said, different sources of the nutrients are important. So what they can digest and what's just available. We incorporate some fish roe when it's available, and we see big differences in diets with the different type of the roe that we use. So different sources of the nutrients is critical.
So I remember Joe Lichtenbert, who you may recall, Reef Propagations Inc. in Chicago. One of the first basement clownfish breeders. He would feed sushi masago, and he was just, I think he would pick it up at the Asian markets and would just like scoop it out of like a deli container, and he would just go straight into his tanks. But he was always good fish eggs in, fish eggs out, fish eggs in, fish eggs out. That was his thing. But what kind of fish roe do you use? Where do you get it?
We get whatever we can find because it's not always very available. But mullet roe is a common one that we use. We've used some kingfish. What else, Fred? Mullet roe is the ones we try to keep on stock.
That's about it for actual roe. What we've used in place of roe, the next best thing that I've found that we're already using for other purposes is the same sort of enrichment products that you would be using for your rotifers. So products from Reed Mariculture, Inve, other companies like that. It's basically just a super powdered or liquid condensed goop of high quality fatty acids that were derived from, good marine sources.
Some source of marine,
yeah. But a nice big thing of mullet roe goes a very long way if you can get it.
And being in Florida, mullet roe is probably relatively obtainable, I'm guessing.
Yeah.
All right. Fred, what other things or what other advice would you just have for the home aquarist who's thinking they want to try their hands at marine fish breeding in general?
Know that your setup is not going to look as pretty, and you're going to have to rig all sorts of contraptions out of materials that are not for that purpose. Our egg collectors are made out of 5-gallon buckets and Nitec screen. We have ones that are made out of bread baskets and dog food containers. You're pulling materials from a lot of other industries. You can rely on the pool industry for a lot of stuff. Be good at plumbing before you start setting up an automatic harvest collection device, because if you're bad about your plumbing, you're going to overflow your whole giant barrel of copepods onto the ground. So when I was first getting into this, in my teenage years and working at a pet shop, I was so afraid of everything that was not the pre-assembled Eheim filter with this hose and this clamp and whatever. And then once you get into the industry, you'll see every facility has some crazy rig where you're like, you're using an oil filter pan to do what? And it's like, this is a very crucial, important part. But yes, it is an oil filter pan from AutoZone to make it work.
I have a question for you, and it kind of goes back to that whole thing about the successes, 1, 2, and then 3. Was that after 23 failures, or was there a 1 success and then 5 failures, and then the second, how did that play out? And then also, at this point, now that you've done it, this is a follow-up question to the same. Now that you've done this successfully a few times, 7 times, I don't know, 8 times, 9 times, does that mean now it's easy? Now you got it? You can just punch them out? You can just crank out 1,000 fish?
Two parts to that. The second and third fertile spawns ever were our first and second runs. So our very first spawn was only 67 eggs, and we stocked it, but they just didn't last. So right from the get-go, we had our first successes. I think it's important to point out, when we're going back to talking about the minutia of taking care of these tanks, somebody who works here, Andrew Palau, has raised Centropyge angels before. He's raised lemon peel angels for a private producer in Florida here. So he kind of knew what to expect and guided a lot of time point introductions on things, or just sort of an understanding of, all right, we can count on by day whatever that they'll be ready for this sort of a thing. So that gave us a little bit of more of a familiarity with it. I would say any given large fertile spawn of them, we could pretty much promise that you could raise some fish out of that. Dr. Ohs and I were talking about just today. We have 2 broodstock fish right now that are employed, and they have, just from a couple months of us attempting to raise the spawns, have generated hundreds of half-black angels.
Wow.
So those 2 fish, if you just actively try to raise 6 months worth of their spawns or a year's worth of their spawns, I mean, I don't know what the market saturation is for half-black angels, but I don't think you need to take them out of the wild anymore if you can raise, a couple thousand of them a year from 2 fish.
Yeah. Yeah, I was just wondering, because of that percentage, like your success rate was going up and up. And then it kind of made me compare it to, like, how you have your first kid and you're so careful with that first kid, and you take tons of pictures of the first kid, and then you take the second, you have the second child, and it's like, he'll be okay. And then by the third kid, you're like, whatever. If he survives, he survives, And I was wondering if maybe your breeding successes were picking up momentum because you're just used to what's going to happen, and it's not even that the eggs are better than before. It's more like you're better at what you're doing. I don't know. That's where my brain is going on this thing.
It's a little bit of that. One of the intangibles, just as a little like anecdote, again from Andrew, we might have been adding the same flow of water to the tank at the same rate, but the position of the little tube that's dropping the water into the tank can make a difference, and has made a difference. He'll swear by it, and the producers that he's worked for will swear by it, that certain fish, just what you're doing to the surface of the water, the flow dynamics that you're creating by the angles in which you're introducing water and the speed and everything can make a huge difference. But that's not something that is easily measurable. So day by day you're building on. If you figured out that at day 9 you did something slightly different, then everything younger than that is going to benefit from you making that very minor adjustment.
Yeah. And now day 9 we got to do such and such, and it just becomes a routine.
Exactly, that too. Yeah.
Yeah.
So I'll just circle back to your question about market saturation. I pulled up really quickly on aquariumtradedata.org because that's the only place we really have data so that we can actually point to. It looks like, I'm just going to ballpark and say about 10,000, 12,000 a year on average for the years that there's data of that species being imported into the U.S. So just under 10,000 in 2004, 9,000 in 2005, 14.5 in 2008, 13,000 in 2009, and just over 10,000 in 2011. So yeah, I would think being one of the less expensive Centropyge, it probably is one of the ones that probably is of interest to beginning aquarists. it's one of the ones that's more accessible because someone who's just starting out. I mean, if you want to just a quick comparison, let's just say lemon peel. Let me pull it up here really quick. Flavicima. For the same time period, lemon peel was more like, I would say, just ballparking the average of 15,000. So
Okay.
it's 19, 13, 18, 17, 14. So I'm just going to say 50% higher import rate roughly. Just skimming those numbers. So and it's a little more expensive fish. So yeah, it kind of all makes sense. And a little bit more of a touchy fish. So yeah, it makes sense to me. So yeah, I mean, I think there's reasonable demand for a Pearlscale Angel. Maybe a little more than I would have thought of, to be honest with you, because it's a much
that's way higher than I thought, honestly.
Yeah. But now would you say? I mean, also, I mean, now that doesn't include the price points, and googling price points on fish can get a little tricky. But I think a lemon peel is probably a little more expensive fish overall too. So, I mean, so not to put you guys on the spot, but do you think that this is a fish that could be produced commercially successfully, with what you saw in this experimental phase?
It certainly could, and this would be one of the many species that they were raising. So that's the key: is you have multiple broodstock populations of different species, and they spawn at different times of the year. So you have all of your hatchery production going on, hopefully year round, and so you're using all of your equipment and all of your employees equally. So yes, this would be one of them that you can have regularly spawn for periods of time. Yes.
Matt told me recently that when it comes to breeding fish, that there's 4 classifications: A, B, C, D. That's
from the Marine Breeding Initiative's scaling. That's
okay.
Specific to that.
So I was just curious, where would this one land in the A, B, C, D range?
Well, I'll jump in because they may not be familiar.
Yeah, which one's A and which one's D?
I'm raising A, which is easy.
Yeah, so A is basically direct development species. So like Acanthochromos polyacanthus, Pteropogon caudini, so Banggai cardinalfish, Spiny-chromis, fish that have no pelagic phase of any kind, and then some of the really easy ones, like let's say Ocellaris might have been an A. And then at the other end, basically almost any of the pelagic spawners were Ds.
And this is pelagic, so this could be D,
so this
right?
would have been a D. If someone had, Centropyge bispinosa, coral beauty would have been a D. This would have been a D. This would have been carried much more points in that whole Breeders' Award program and the scoring system. This would have been a Class D. So, but then as a frame of reference, a yellow tang would have also been a Class D. We kind of just looked at all of those would have been Ds at that point when we came up with the scoring. Yeah.
Small fish, you only require a pair. We're consistent spawning on them when we're giving them perfect conditions, and so it is very reasonable compared to a large school of fish needed in larger tank.
So I wanted to circle back because you did mention, Cortney, something about seasonality. Is there any seasonality to this, or is this a year-round spawner?
So I would assume it's a year-round spawner, but they probably all take breaks. And we're in Florida, and we have different seasons. And so the fish here know full moons in a building that they're not exposed to. And so we get a lot of Caribbean angelfish spawn at full moons. And how do they know? Because they're not, but all of it's still cyclical. And yes, this would be consistent tropical type of temperatures, but changes in temperatures and seasonality changes alter their nutrition composition and what they consume, and sometimes they need breaks as well. Different populations, Pacific blue tangs would almost spawn every single day for us, but they're over a period of time, their egg quality would slightly go down, and that was a nutritional thing that we couldn't necessarily make back up. But we know that now, and so you can make it up.
Do you purposely give them a break or force them to take a break to prevent them from kind of burning out?
that can be a strategy used as well, and for just the number that you really need and what your purpose is for research versus production and how many you need to produce. Yes, that 1 degree Celsius can make a whole lot of difference.
Matt, I don't understand. You're saying that too much sex leads to burnout?
I mean, yeah. I mean, I don't know. I mean, like breed yourself to death. It does happen, I guess. So.
All right, guys. I want to show you our upcoming cover. I've got this slide ready for you. This is the new issue that's coming out. Scoot over a little bit there.
It's a better picture than I could ever take of them.
Okay, let's talk about that for a second. I have been super crazy picky about the pictures we get from breeders. Like, come on, do better, And then when I started to raise these microscopic things behind me, I was like, oh my God, this is so hard. And I finally appreciate what you're going through. And I was like, all I want is one good photo. You've got every day of your life to get this shot. So anyway, I happened to get this one amazing picture that I probably will use in my editor's letter, Matt, where I took a picture of a fish. I want to call it a fry, even though he said it's not a fry. That was
yours is a fry.
It is a fry?
Yours
okay. It hatched, and it was minutes old. I mean, it literally just emerged from its egg, and I happened to see the squiggle moving in the water change water, and I took a little tiny container, I scooped it, and I put it down on the floor, and I grabbed my macro lens on my camera, and I've got my cell phone on the side with the flashlight turned on to light the subject, and I'm trying to photograph this little tiny thing that keeps swimming out of focus, and it keeps swimming away from me, and then it swims back into the frame, and I finally got this one amazing picture. If I had it handy, I'd show it to you, but I don't have it handy.
Well, maybe we'll close out this interview. I won't let we'll play a guessing game, but I won't let Cortney or Fred actually guess vocally. They can send me their guesses afterwards. So we'll fill them in on what's going on here. So the deal is, I had someone send Mark some fish eggs. He put them in a 10-gallon tank. He doesn't know what he's rearing.
Right.
So while he's digging up a few photos, he can show you the latest photos he posted. So he can show you some pictures of the eggs. He can show you the picture of this baby fish. You guys may or may not know what he's rearing. Just don't say it if you know, because he doesn't know. The deal was, he has to rear them.
That's my photo.
He can share as much info as he would like about the conditions and how he's doing it, but he does not know what species of fish he's rearing. I will say this: Mark hates freshwater fish with a passion, and he and I, for like 20 years as friends, long before we brought him on as the new executive editor for Coral a couple years ago, he and I would send I have no qualms. I don't care about salinity. I don't discriminate. Mark, I don't know. He's kind of a salinity racist. I don't know. So he'll send me a saltwater fish, and I'll send him back a beautiful discus, and he'll send me another angelfish, and we'll have this war of salinity. So he's got these eggs. He's got these fish that he's rearing. Did you pull up some of the egg pictures? Go grab a couple more.
Yeah.
So, and tell him what you're doing. And there's a couple things you're not allowed to say. But right. So he's been sharing this journey on his YouTube channel. He's officially and now his fish that he's reared are already laying eggs.
These are the new eggs.
Yeah, and so good eggs and bad eggs right there.
Yeah.
He still doesn't know what he's rearing.
Right. Here. So anyway, that one picture I showed you first, that one was 3 millimeters long. Those are brine shrimp egg casings, or Artemia, and this little guy was just I couldn't believe I got the shot. I was so excited. It's fully in focus. I'm so proud of myself, and the thing is minutes old. It's just the tiniest little squiggle of life. Here, I'll show you a picture of what the fish looks like.
Okay.
If I can find one. I keep finding egg pictures because they don't move. They're so easy. Let's see.
So I think it did give this project gave Mark a new appreciation for the challenges we all face. So those are, I think, the oldest pictures he has. So
yeah, this
you
is
guys
from,
might recognize them.
October 21st.
So you guys might recognize what those are. You might not. Google Lens did not figure out what those were. Mark hasn't recognized them yet.
No.
So don't say what they are if you know. We've been calling them saltwater crocodiles because I shipped them a bunch of eggs. Mark, you want to explain what they're living in right now?
This is what I'm making. Okay, so what I'm doing is I'm just raising them. Well, I don't know, Matt, if I'm going the direction you wanted, but the eggs were sent to me, and the first day, 3 of the fish emerged from their shells and were swimming around, which was cool. And then over the next 15, maybe 20 days, they all hatched, and I had 100% success rate. So I don't know why it's so hard for you guys. I had 59 of these little fish swimming around. And fast forward about 60 days, I saw these little orbs in the bottom of my tank, and I was like, what are these? That's weird. And Matt just starts laughing, and I'm just like, what? And he was like, those are eggs. I'm like, I'm raising fish. I don't have time for this all over again, you know, because I'm growing baby brine shrimp every single day of my life now, which is, I'm sure it's 10 times or 100 times easier than your rotifer and your phyto and your copepod stuff you're doing. But anyway, I still have to do this every single day of my life as a new extra task. And I've been feeding these fish, trying to grow them to the point where I can recognize what they are and identify them. And they're busy making more babies. And so I was just like, well, I'm not going to care about it. But then I see the little guy at the top, the little black fish, 2 eyes, little squiggly tail. I'm like, aw. So I ended up, like I said earlier in this thing, I poured some water change water through that breeder basket and clipped that in the back of the tank. And now I've got like 18 more in the breeder basket that are really tiny that I'm feeding them baby brine. I'm feeding the tank baby brine. And it's just been an ongoing thing. So the tank's behind me, but it's turned off right now. It's dark. But yeah, I've got 70 fish so far, probably, give or take, that I'm raising. And apparently it's never going to stop.
About the eggs?
Yeah.
We've made Mark a fish breeder. I made Mark a fish breeder. He's a fish breeder now, guys. Like,
he said, because he said I went all the way through from egg to fish to egg to fish. Yeah. He says it's official. You've actually done it.
Close the life cycle.
Yes, that's what he
said.
Life
cycle.
Yeah.
And he still doesn't know what they are. I love it. It's hysterical.
Did those fish eggs initially arrive in water, or did they arrive in some sort of thing like wet sphagnum moss and air?
They arrived
Can you say that?
Not in water.
Okay.
They did arrive in something. That's a really good guess, actually.
That's a big hint for everybody at home.
Should I tell him what it was, Matt? I mean, I don't care. I mean, you can
tell him. It's the cat's out of the bag here. Yeah.
Yeah.
You can tell them how they were shipped, sure.
Yeah, so it arrived in a mop.
Mm-hmm.
Like a fish mop. And
yeah.
And matter of fact, after...
Was the mop in water or no?
Just a tablespoon of water, maybe. It was mostly a bag of air.
Okay. I didn't realize that. I thought the mop was submerged, but...
No, I mean, I don't remember. I'd have to look at my photograph again, but I remember as a matter of fact, the thing floated on the surface of the tank. And I was like, how do I temperature acclimate this thing? Because it's so floaty. And he arrived during the heat of Texas summer, so it was pretty warm.
Yeah, I thought he shipped it submerged, but I didn't realize he didn't put water on that mop. But
yeah.
I think Cortney in this lab raised a similar thing, but different family, but a similar kind of thing back in the day.
Mm-hmm.
Yeah, it's once Mark figures it out, it'll be very funny. It's a little it'll be I'll just chuckle a whole lot. I mean, I've been laughing the whole time. It's hysterical to me, but
he's setting me up. That's all there is to it.
Yeah.
So,
if you know, you know, and at this point, a couple of our friends know what they are, and so it's been really funny to. And those are not fully developed. That's the best part. They're going to look even better. So
that's my breeder box right there, and there's some little tiny black dots in there. And then, of course, the main fish are in this other picture here. Give me a second here. I'll find it for you guys. Since you guys are into breeding, you probably like this stuff.
And so we also set them up with a matin filter, which is my partner, Stefan, runs Swiss Tropicals and does the matin filters. So instead of just a typical sponge filter, I was like, Mark needs to have a matin filter. Like, needs to understand what that is. So, yeah. And then, so we used a matin filter, so that wall sponge instead of just a little,
I have all these videos. That's the problem. I don't think you guys want to watch a video. But anyway, here's those eggs I captured.
Yeah.
And you can see a whole clunk right there in the middle, But look at all these loose ones that I found too, So they ended up in the basket. That's why the basket looked dirty. But you saw the one picture
nice.
of the fish earlier. I'm scrolling through my phone right now.
Oh, and what salinity are you doing them at, Mark?
1.017.
So it's 2/3, two buckets, or two parts salt, 1 part fresh.
Howdy.
I'm gonna mop in here.
No, we're good.
All right, okay.
All right.
I'll come back tomorrow then.
Okay. Thank you.
Oh,
Here's
there's
another
a nice.
one.
That's a nice photo. I haven't seen that one.
That's a little one.
Hey, that's good.
But I was trying to find you just a group shot. I mean, these are not great pictures. Oh, man, you can't see anything. That's why you have to use a Nikon.
But that's that. Yeah, I think that was a great learning experience for you. Just to like, I remember having to tell James Lawrence, like, you got to give these breeders a break. This is really small stuff.
Yeah. Hey,
They're doing
he's
the
right.
best they can. All right, well,
I'm trying to
guys,
bring
if
us
you
back.
want to message me your guesses, I'll tell you if you're right. But Mark's not allowed to tell Mark.
Yeah, the idea was that hopefully I can identify it. I might never identify it. I mean, I'm a
you'll at
coral
some
guy
point you will.
more than a fish guy, but you know, the guy that
some
sent me
point-
the eggs said if you would just Google my name, you'd know what these are. And so I've not Googled his name. But Matt did say only a handful of people have raised these, which that's kind of curious to me.
They're not common. They're not common in the hobby or the trade. They are pretty rare. So
Okay.
So that makes it
Well, that
more
changes the answer.
intriguing, you know?
it was interesting, and it's interesting because some of the guesses Todd Gardner gave me a really good guess. It was like that. I am shocked that was your guess, and you're absolutely wrong. But that is a really interesting guess. That was that. Yeah, Todd had a really interesting guess. So it sparked a lot of interesting conversation. So it's been a really fun project. So
Yeah.
yeah.
So yeah,
So
now I
any
kind
last-
of relate to you guys a tiny fraction of a bit, but I don't want to get a PhD in fish raising or anything like that. That's let Matt do that. He can have my slot.
So Fred or Courtney, any last words here before we let you guys get about your day?
If you
You've
have
said it
questions,
all.
let us know. We're
No-
here to
no-
help. So we want to
no-
see this
no
everybody succeed. So that's what we're here for.
All right. Come to University of Florida. It's a great place to study. Or
Well, that, yeah. And, just for if you're trying to get into this industry, just get as involved in whatever animal stuff is available to you. And that will open doors for you, I would say. When I was at a pet store reading about Dr. Chad Callan working with the yellow tang, I was like, like I want to, do something like that someday. And I didn't get to where I'm at by going right into raising yellow tangs. I had to, work with wrangling alligators, and I had to work with sea urchins, and I had to scrub a whole bunch of tanks and just find little inns where you can, even if your program or your school is not necessarily the University of Florida's, master's aquaculture program. There's something that you can find to further your education and get your foot in places and get dirty. Be prepared to get very dirty.
and stinky. It is smelly work. It is.
Yeah.
All right. Well, I really appreciate your time, guys. Thank you very much.
That's what I was going to say. I said thank you both for taking the time out of your day to discuss everything with us and kind of give us extra information behind the scenes. It does help build a better story, and we appreciate the input. We're really glad that you're willing to share this. That you're willing to be open about it, because it's very practical knowledge, and hopefully those that do look at these stories, when they read them, think, I'm going to try to see if I can do this in my own home tank. I mean, that's the whole point. And if an aquaculture business sees it and learns from it, okay, great. It's still another source of bringing fish into the hobby, which is great. So I love all that.
Exactly.
So, Mark, that cover one last time. You want to show it and tell everyone what issue this is going to be in,
Yes.
or is in by the time this.
So this is our January-February issue of Coral Magazine, Breeding Breakthroughs, and that is a half-black angelfish on the cover. It's beautiful, and I'm very excited that we have that here. So I appreciate you guys helping to... So this cover right here actually was my inception. I was wanting something on the front cover, and I specifically wanted a fish that was being bred, and then I wanted the background to be something egg-like. And that's why we went with those tunicates, because of their shape of their bodies. It was just like... And it was blue, and I really wanted blue on the cover desperately for some reason. I was like, had to be blue. So we came
So,
up
Mark,
with that.
That's why you're wearing a blue shirt today.
I am.
Mark, what year? You said January, February, but what year? Coral's been around
2026.
many years.
2026.
All right, there we go. January, February 2026.
Yeah, this is volume
So,
23.1. So this
Yeah.
magazine's been around 23 years.
Yeah, we're starting our 23rd year.
Wow.
And
Yeah.
if you're a Coral subscriber, it will be in the digital archives, so you can read it digitally. If you didn't get a print copy, or
Yeah.
if you subscribed after that issue, it'll be available as a back issue purchase in the store once it's online. If you're shopping your local store and they carry Coral during January and February of 2026, buy a print copy there. Support your local store. Again, Fred, Courtney, thank you for joining us and giving us your time and sharing your research and your findings, and for elevating aquaculture within the aquarium trade and the hobby. It's really important, and I'm really thankful for your friendship. And Fred, it's been a pleasure to meet you and talk to you today.
Thank you both for everything you do for the magazine.
Oh, absolutely.
the whole trade.
We love it. It's
Yes.
it's a passion project, that we can't stop doing, and every single time we finish one, we start the next one. I have a question for you. Did
Go.
Matt told me
Why?
that with the
'Cause
fish?
I'm in an interview right now, dude.
What's happening?
Sorry.
All good. All good.
It's okay. Did you just scream, "Get out"?
Yeah. That's our guy Freddie. He's worked in aquaculture forever. He's a
Oh
good
my God,
dude.
2 Freds in the same place. That
I
must be rough.
know, and it's
Oh.
that's the aquaculture is the only place I meet other people named Fred, and they're all way older than me because
Oh, that's
nobody
funny.
names their kids Fred anymore.
It'll
Okay.
come back around. It'll come
My-
back around.
My question, it's not a hard one. I just